Well, yeah, wish if everything is fine. Now we'll come to lecture 8, and lecture 8 actually have 3 parts because it includes a very important topic, which is how to color proteins, RNA, and DNA residues and sequences in the alignment using Jalview. So you will see many coloring scheme, and we will try to describe the difference between each one of them and why we can use each one of them and in which cases. Jalview color scheme, as we said earlier, it has many coloring scheme. It can be used within Jalview. And 3 of them actually are dynamic and use conservation and consensus analysis to determine the color. So these are the 3 are the percentage identity color scheme and cluster X coloring scheme and Blossom 62 coloring scheme. Of course, Jalview color scheme are rendered behind any sequence feature, and that's colors. That's why we have to keep this in mind. So it may be necessary to disable those features. So before you choose your color scheme and apply your color scheme, you have to disable first the feature, the sequence feature, because it will have its own color. So that's why we have to disable them first. Then we have to color our alignment. So how we can disable it? We just simply go to the view menu at arrow number 1, and then go to show sequence feature at arrow number 2, and just press on the show sequence feature. Just check this one, uncheck it. Uncheck it means so you will not see this arrow here. So you will not see at arrow number 2. Just uncheck this. Just remove this sign. So when you uncheck this, this means that it's not sequence features is not applied anymore. Okay, we'll go here to how to load alignment using the steps demonstrated in the previous video from the BFAM-C database. Let's see how we can do this. Simply, we can fetch any sequences as we can demonstrate here. First, you have to go to the file menu, and from file menu, you just go and at arrow number 2 and just press at arrow number 2 fetch sequence. When you fetch sequence, you will get this menu. That's pop-up menu here will appear to select the database which you will choose from. So as arrow number 3 shows, you just press here. So you will get this drop-down menu here, and just choose PFAM in this example, for example, and then put the accession number that belong to this database. So I will choose this PFAM accession number, which is PF03760, which appear here at arrow number 5. And then you can press OK. If you have more than one sequence, if you would have a big multiple sequence alignment, you can put more. Just separate them by semicolon, as we said before. So you can choose 1, 2, 3, whatever, 10. So you will have a multiple sequence alignment sequences. We just put here as many sequences as we can that needed for our research, and then separate by semicolon, and then press OK, as we can see. Now we would like, after making, of course, importing the sequences and making our alignment using one of the program that we studied in earlier videos, cluster Omega or cluster W or NAFT or TCAFE. After making the multiple sequence alignment that we need, we have to be sure that it's not colored. So we have it like this in front of us here. We need to be sure, as we said, it's not colored, and we need to color it now. So first of all, we will go to the color menu here at arrow number 1, and choose the first choice in the menu, which called apply color to all groups. So we will unselect here this also to be sure it's not selected at arrow number 2, that this first option is not selected. We have to be sure that it's not selected. And then we'll go to the second option here, which is called none. None means that the coloring, there is no color. So we have to make sure that apply color to all groups is not selected. And then we'll go to this option at arrow number 2, none. None means that no coloring is selected now. Now, after that, we will just go and choose the color we want. First choice we will have is cluster X. Cluster X is a very popular coloring scheme based on the default coloring scheme of cluster X program, as you can see. And of course, you can get more information about the cluster X coloring scheme, because this is an emulation of the default color scheme used for the alignment in cluster X. And what is cluster X? Cluster X is a graphical interface for cluster W, multiple sequence alignment program. Okay. What we will see in this cluster X, we'll see that each residue in the alignment is assigned a color if the amino acid profile of the alignment at that position meets minimum criteria specific for residue type. This means that there is to be colored, the column to be colored, it need to meet a specific criteria. So we will show you an example for that. The example for that here, if you can see, for example, the amino acid K and amino acid R is colored red in the column if the column includes more than 60% K or R combined, or more than 80% K, R, or Q individually. Okay, let's see how this apply. This, for example, we have the red color. This means that this category of amino acids are positive charge. So here we have the K and R, as we said before. These two amino acids, if the column, if it contains K and R more than 60%, these two amino acids, more than 60% of the content of this column, it will be colored red. If it have 80% K or R or Q individually, it also will be color red. The same thing apply for other colors. So as you can see that this criteria always just apply according to here this threshold residue group, as you can see here. The other way of coloring may be much easier. It's called Zappo colors. And Zappo colors, the residue are colored according to their physicochemical properties, or physical and chemical properties, as follows. If the amino acids are aliphatic hydrophobic amino acid like ILVAM, like these ones, so in this case it will be colored this color. If it's aromatic, it will be like FWY, it will color this one. If it's positive, will have this blue color. If it's negative, like D and E amino acids, it will color the red color. So each color have physical and chemical properties, unique physical and chemical properties, so it will have also its unique color, as you can see from this table. And this is very easy one. Tailor is another way to color your amino acids in the multiple sequence alignment. As you can see, it's invited by Willie Taylor. That's the name of the scientist who invite this coloring scheme, and that's why it's called Taylor. And of course, a lot of description about it is available on the book here, and this reference here, Protein Engineering, Volume 10. And actually, as you can see, each amino acid have its distinct color, so it's easy way to differentiate and understand the way it works. Also, we have some coloring scheme generated by GCOS software. So this color scheme called Flower was generated by GCOS software, and actually Flower coloring scheme is a light color scheme based on the Blossom 62, as you can see here. Also, you have also another one called Blossom, and this Blossom one is color scheme also generated by the same software. It's called GCOS software, and published in PMC Bioinformatics in 2020. And as you can see here, this one is called Blossom. It's a light color scheme with high contrast based on the Blossom 62 also. As you can see here, each amino acid is individually colored, as you can see here. Also, we have the same, also from this group, we have also Sunset. It's called Sunset. This is also a coloring scheme, also generated by GCOS software, and the same reference here. So you can see here, it's Sunset is a red-green color vision, deficiency adapted color scheme based on the Blossom 62 also. So this color scheme, the three of them are new actually, and you can find it easily in and recently in Jalview. Also, the number 4 from that group is Ocean. Also, Ocean color scheme generated also by JCOS software at the same reference here, at the same year. So the Ocean is a blue shifted light color scheme based on also Blossom 62. So this 4 of them, you can read about them here at this reference available here mentioned in the slide. Okay, and now we'll go to a different one, a little bit different from the others. It's called hydrophobicity. And this is, of course, the coloring will be based on the hydrophobicity of the residue. So according to this hydrophobicity table, in the from published in Kite and Doolittle, you will see that the most hydrophobic residues, according to the table, are the colored red, and the most hydrophilic is colored blue. So as you can see, all the amino acids are here. The most here, the most hydrophobic here with the red one, and the most hydrophobic are the most colored blue one. So, and you have these two extremities, and you have the range of other colors in between. And here is helix propensity. This helix propensity also the same thing. The amino acid that involved mostly in making the helix having this color and the other one having that color. So and you have in between them the range of it. So you can see the contrast in these specific characters. And then also you have this turn propensity so you have also these two extremes can be seen, and the range in between them as you can see here according to the involvement of this amino acid in the structure of turn propensity. And this is paired index, the same thing apply here. As you can see, the last few ones that we described always like concerned about the structure of the and how is your amino acid involved in the structure of your protein. Also, you have here the two extremes like this. See the most blue here and dark blue, and then here dark green is here at the other extremity. So these are the amino acid and the way you can color them. Now we'll come to the nucleotide colors, and this will be very simple. That's why we left it in the end. You have here the nucleotide colors. You have 4 nucleotides. Adenine is green, and cytosine is yellow, and here red is guanine, and of course thymine and uracil are blue. Of course, because they are the same group, and a little bit some this for DNA and this for RNA. Both of them are blue, light blue. Here, purine pyrimidine colors, you can see here that purines A, J, or which is R, represent A and G. These purines are with this color, and the pyrimidine for that with that color. So this is represented with this, and that these group are represented with that. So only 2 colors, so it's easy to understand and comprehend. Blossom 62, as we said before, it's a dynamic one, coloring scheme. As you can see, gaps are colored white. If a residue matches the consensus, sequence residues are the position, it's colored dark blue. So we have the gaps are white, and if the residue matches the consensus, it's dark blue. And if not match the consensus with residues, but 2 residues have a positive blossom score, it's colored light blue, as you can see here. So if it's dark blue, this means that it matches the consensus, the colors, and if not, but 2 residues have positive color scheme score, it's colored light blue. So you can see that some of the columns are light blue and some of them are dark blue, as you can see here. So it's this only blue, light and dark blue, as you can see here. This column Blossom 62 score. Also, blue can be used in the PID coloring scheme.
and by it's called by percent identity. The BID option for the current schemed residues, as you can see here, according to the percentage of the residue in the column that agree with the also with the consensus. Only the residue that agree with the consensus residue for each column are colored. But we have now 4 colors can be used, or for like you said, not 4 colors, but like ranges of the blue color. So if you have this color, which is white, so this means that less than 40% of the residues in that column are agreed with the consensus. Here, this color a little bit darker, which means that the residues in that column agree with the more than 40%, actually from 40 to 60 consensus, agree with the consensus for each column as covered here. Here, if it's more like from 60 to 80, it will be more than 60%. It will be at that time, it will take this range of color. Also, if it's a little bit darker like this one here, this means that the column here, for example, more than 80% of the residues in that column agree with the consensus. So that's why a little it's more dark actually, as you can see here. So by just looking at the range of that blue color, you can know if it's dark blue or light blue or much lighter blue or white. You can know the degree of conservation in each column, and this is very easy way to comprehend and to differentiate between columns. It's called PID, or percent identity coloring scheme. Okay. This is only practical. You can just go ahead and do some practice here, or go to this multiple questions here. You can just go and open this link, and of course you can answer the question to practice with the content of that lecture. Thank you very much, and seeing you in next lecture. Man, see you. Bye bye.